Peace and blessings. Welcome back to another video. In this video, we are going to discuss again about the process of transcription. So in previous video, we discussed about the process of transcription and compared the transcription in prokaryotes and eukaryotes. So let's again recapitulate. In case of prokaryotes, we have seen DNA is transcribed into the mRNA and then the mRNA is translated into the protein. But when the same machinery is recapitulated in the eukaryotes, we see that this whole mechanism it becomes more complex. Let's understand. So again, in case of eukaryotes, we have a DNA, and from the DNA we have the process of transcription, whereby a primary RNA transcript is synthesized. But the RNA transcript it's not directly then translated into the protein. Rather, it undergoes a series of reactions. We call that as the RNA processing. So it includes the 5' capping, RNA splicing, and 3' polyadenylation. So the transcript, which is synthesized from the DNA, it undergoes the process of RNA splicing, where its 5' end is capped and a poly A tail, that means the adenine nucleotide tail, it is attached at its 3' end and it undergoes the RNA splicing, where the non-coding regions they are removed and the coding regions they are joined together in order to synthesize a protein. So we will understand the RNA processing and we will take all of these steps one by one. Before that, so let's again take a look what we discussed earlier about the process of transcription. So now we know the structure. So this is an RNA polymerase which is attached on the DNA here. And the RNA polymerase, it has a tail. We call this tail as the C-terminal domain. So the C-terminal domain, which we have studied earlier, it has certain sites. Like we studied about the 5' site, which contains serine, and this phosphorylation at that particular site it helps in the initiation. Now this also helps in the recruitment of the capping proteins. If you see here, in the next step, the RNA is synthesized and the phosphorylation at the 5' position, it is also aiding the recruitment of splicing proteins here. In the next step, you can see that this is also the same phosphorylation at the 2nd and the 5 position. It is also aiding the recruitment of the proteins which are involved in the 3' end processing. So that means which will be involved in the formation of the poly A tail at the 3' end. So those protein, all of those proteins which are involved in the RNA processing, they are recruited. So this C-terminal domain, in a way, it serves as a landing platform. So you can see how important or how crucial the phosphorylation at these sites is. Now let's discuss the formation of the proteins. If we talk about the prokaryotes, so the same gene sequence, it can give rise to, so if suppose this is a coding sequence, the same region of the mRNA, it can give rise to multiple proteins. It may be a protein alpha, a protein beta, or a protein gamma. But in case of the eukaryotes, one single mRNA, it will code for just one protein. So in fact, when we talk about prokaryotes, we say that it is polycistronic mRNA, and when we talk about eukaryotes, we say that it is monocistronic. So it codes for just one single protein. Now let's understand. So we will take all of these RNA processing steps one by one. In this video, we're going to discuss about the 5' capping. So let's understand this reaction first. So in the capping, it requires three enzymes. The first one is RNA triphosphatase, the second one is guanylyl transferase, and third is methyl transferase. As you can see in the first step, so this is the transcript, RNA transcript, which has been synthesized, and this is the 5' end of the nascent RNA transcript. And first and foremost step, that is catalyzed by the RNA triphosphatase. So this bond between the beta and the gamma phosphate, it's broken down, and this beta, it attacks the alpha phosphate of the incoming guanosine triphosphate. And now we can see that this guanosine monophosphate, because the two phosphates they are removed, so you can see here in this, the inorganic phosphate is removed, and the guanosine monophosphate, it is now linked to the RNA transcript, and it is linked on the 5' end of the transcript. So this reaction is catalyzed by guanylyl transferase. Then the next step, the methyl transferase enzyme, which catalyzes the transfer of methyl group from the substrate is the SAM, that is the S-adenosylmethionine. So this methyl group, it is attached at the N7 position of the guanine. So this is how the capping occurs. So these reactions, they basically signify the capping at the 5' end of each RNA molecule, which are synthesized by the RNA polymerase second. Now, this is called as a cap zero structure. And if there occurs any methyl transfer or methylation at the next base, that we call as the cap 1. And it has been seen that if the adenine is if the methyl transfer occurs at the second base after this guanine, it occurs at the N6 position. So first it occurred at N7, next it occurs at N6th position. So this was how the capping occurs. Now you may say that why the RNA transcript needs the capping? Now it has various functions. If we say this methyl cap, it basically signifies the 5' end of the eukaryotic mRNA. And this landmark, it basically helps us in order to distinguish the mRNAs which are synthesized from the other type of RNA molecules which are there in the cell. For example, the RNA polymerase first and RNA polymerase third, they do not have the C-terminal domain. So that means their polymerases, they lack the C-terminal domain. So it helps the future mRNA be further processed and exported, which we saw in case of the RNA polymerase second. It also has, now this 5' end, the cap end, it also has a very important function because it prevents against the function of nucleases. So it prevents the degradation of the transcript which is synthesized. It also helps in the transport because once synthesized, it has to be transported to the cytosol for the synthesis of proteins. It helps in that transport. It also helps for the recruitment of those proteins which we will discuss ahead, which help in the splicing and which also help in the 3' tailing. So this was the significance of capping, and we discussed about how the capping basically happens. And next we will take the process of splicing. Hope you have understood the concept well.